Okay, thank you. Good morning. I'm Tim Parrott, and I work in the information management group at Dart Neuroscience. And I'm going to be talking about an end user tool that we created for designing chemical libraries. So just a little background. Dart is a privately held company in San Diego focused on memory and cognition. There are about 200 scientists at Dart, and about a quarter of them are chemists. And a couple years ago, a new group was formed focused on parallel synthesis to assist our projects. And right now it's staffed up to about 20 chemists involved in various phases of chemical library design and creation. And that group came to the information management group and said, we need a chemical library design tool. And so that is the impetus for what I'm going to show you. So design is just one component of the discovery paradigm. And as such, it sort of puts some natural boundaries on what this tool is going to do. Excuse me. So it's not going to do analysis, and it's not going to interface directly with synthesis, but it does need to be informed by the results of maybe HTS analysis. And then the key steps that the design tool needs to do is a user needs to be able to select reactants, enumerate virtual products, calculate properties, and score these virtual products against our computational models, and then have some way of analyzing and filtering all these potential compounds and coming up with a set of something that they really want to do. And ideally, the tool would put something out that would be beneficial to the synthesis part. So that's the basic framework. There were some constraints. We are a fairly small IM group, and we are a rapidly growing company with a lot of groups with unmet needs. So we wanted to get something out quickly because the users didn't have anything, and we wanted to put something out that wouldn't require a lot of training, because the chemists were kind of already up to here with new software, and so they needed things that they could understand and go with. And the approach then was that we had some existing services that returned things like standard property calculations, and we wanted to build off of that, extend those services and build some new ones that delivered reactions. We identified input and outputs between different programs as one of the bottlenecks, and something that caused a lot of stress from the end users, and so we wanted to address that. And then we were going to move the capabilities to our servers so that we could do some more in-depth calculations that people might not be able to do locally. And the basic equation that we were aiming for is if we made it easy enough to use, we wouldn't have to spend very much time doing support, and then both the users and the IM group would be more productive and happy. And a lot of what I'm going to show you is about ease of use. It's things that we did to make these tools play better and sort of minimize points where the users might stumble. So for platforms, we needed platforms to do some visualization of the results. I mentioned 3D scoring, property calculations. I don't think it's going to be any surprise that we chose ChemAxon. We were already using ChemAxon and had services to deliver standardized properties. Likewise, we were already invested in Spotfire and OpenEye to do some of those calculations. So the really new part here was that we needed something to bring these together, and the data pipelining that we chose was KNIME, and then Infocom comes along as kind of the glue to help ChemAxon play well with KNIME. So these are the platforms that we're going to develop with, and here's what the tool looks like, and this is pretty much what KNIME looks like when you open it up. But there's three things on here that show some of the more visible enhancements that we made. And the first of these is that at the very top of the node repository, there's a collection of Dart Neuroscience nodes. And these are all custom nodes that were written. And it's everything that's needed to do a basic library design. So the users don't need to go out and find any other nodes anywhere else. They can do it start to finish with this collection of nodes. And I'll talk a little bit more about those. And in addition to the nodes, there's a node inspector panel over here, which allows for browsing through some of the options that they have and selecting and configuring these various nodes. And finally, there's a Spotfire export. So once the library design is completed, they can do some of their analysis and filtering in Spotfire before outputting the final design. And so I'll talk about each of these three sections in the upcoming slides. So your library design, the first step is enumeration, and you're going to need to start with some reactants, or in our local parlance, diversity elements, and just drag a node onto the workspace. And then over in the node inspector window, we have a drop-down where people can choose from curated lists of reactants. And once they select the one that they have, those structures come in along with the assigned reactant ID and some information about the stereochemistry of those reactants. So in this browser window, users can go through and select individual reactants, or they can select all of the reactants. They can import additional compounds to append to this list, or they can go ahead and click on the funnel to bring up a Marvin sketch window where they can draw substructures and filter the results by substructure. So that's one of the basic inputs. The other common key case is that people have already done some analysis, maybe from some screening data, and they have a set of reactants that they need to bring in. And so we have a simple node that simply takes a list of reactant IDs and pulls in the structures, and then they can be used further downstream in the enumeration. So for each of our reactants, each salt form is assigned a different reagent number. This is because chemists use benzylamine and benzylamine hydrochloride differently, which is good to store them separately in inventory. It's bad if you're enumerating a library because you'll get duplicate products. And so one of the first things we needed to do was to deduplicate at the parent level. And we built a little node that takes the input structures and uses some JChem magic to deduplicate at the parent level, and it outputs unique parent structures with alternatives. Once you have your reactions selected, or your reagent selected, the next thing to do is to select some of your reactions. And we have reactor nodes that look very much like the ChemAxon Infocom nodes, but there are some key differences, as I'll explain in a minute. But once the bioreactor node is pulled over, a window pops up, and it's a browser window where the users can see all of the available reactions. There's examples of each reaction, and clicking on the examples opens up a wiki page where there's more information about the selectivity of the reaction. One of the things that we identified when we just initially put the Infocom nodes in the hands of some of the users is that people who didn't use it very often kept on getting burned by not getting the correct order of reactants going into the node because it's required ordering. And so the nodes that we built do not have specified ordering, and in fact they sniff out the incoming reactants to figure out which one is the aldehyde and which one is the amine in this case. And so that saves a lot of grief on the user's part. As I mentioned, these nodes are not exactly the same as what you get out of the Infocom nodes. Some of our reactor nodes actually have small workflows underneath them. So in this particular example, it's a deprotection reaction, and what we found is that the people wanted to get everything that went through that out whether or not it reacted or not, all in the same product column, which normally wouldn't happen without a little manipulation. And furthermore, they wanted to know how many of these Bach groups had been removed. And so just by creating a simple little workflow and putting it underneath this, we're able to add some enhanced capability to each of these reactions. Once you have your structures in hand, it's time to get some calculations. And as I mentioned, we had a service set up to return standard ChemAxon properties if it was presented with a structure. And so one of the first nodes we wrote was one to go out and get those properties and allow users to pick which ones they wanted to receive. So it just sends the structures off to the server, and the list of properties come back. There we go. For some of our 3D work, we use OpenEye's rapid overlay of chemical structures, or ROCS. The ROCS nodes, the user comes in and picks a target or a collection of targets, and then within each one of those targets, there's a series of queries that have been deposited there by our computational chemists. And then once they pick the model, they can run the node, or if they need more information, there's a wiki link that goes out and pulls back more information about what that set of queries does. The ROCS calculation returns scores, which can be used further downstream in the filtering, or you can also, we have a way of opening up or launching Vita directly from the ROCS node, and they can view the results that way too. We had a request for clustering. They wanted to cluster both reactants and product structures in order to make sure that they didn't oversample from specific regions of chemical space. And so this node allows them to pick from various clustering methods. And because it's k-means, there's just one cluster, or I mean, they have the ability to specify the number of clusters there. This is another example of a node that runs a small workflow behind it. In this case, we're using the ChemAxon fingerprints and the KNIME k-means clustering. And the output that's returned is the cluster ID for each cluster, and then the cluster size. And that's just carried along in the workflow. So both ROCS and the clustering, and even sometimes the enumerations, can be fairly computationally intensive. And so it's one of the key reasons that we move things to server-side execution is the extra power that you can get there. In addition, we needed to reuse the existing services that we had. And our methods group was able to get some of these calculations to run in parallel across multiple processors, which brought down the time. They actually ended up building a queuing system to handle the different chunks during that parallelization process. And our users routinely enumerate libraries with more than 100,000 compounds, and so you can imagine that some of these calculations take a while, particularly the ROCS calculations, even with the parallelized stuff, can still take more than a day. When we initially had this out there, what that meant is that they needed to be connected to the server the entire time, and if there was, if something got dropped in the network, then they needed to start over. So we found a way to pause the local execution while the server-side execution is still going. And so in addition to your regular run button, there's also a pause button, and then once it's paused, this is the resume button over here. And the way this would work from a node standpoint is that when you're ready to run, you would just hit execute. It would give you your usual, reaction is going, and after a little while, it would change color here, and you'd be able to pause the execution. you get a little warning triangle on your node. And at this point, you can save your workflow, open up a new KNIME workflow and do something else, or you can shut down your computer and go to a meeting or go home for the weekend. And when you come back and hook back up to the network, if it's done, it'll bring the results back. If it's not done, it'll still be doing this, and you can pause it again and come back and check in a few hours. And so people did really appreciate the ability to sort of stop things and go away. Once you have all the calculations done, it's time to do some sort of filtering and selections from that. And our tool of choice on that is Spotfire. So the export to Spotfire is a two-step process. First, an export for a Spotfire node is added at the end of the workflow. And once they're ready to do the analysis, the button up here on the KNIME toolbar is pushed, and it pushes the data into Spotfire. And it takes everything in. So all the calculations, all the data gets moved over, and you can do all the wonderful things in Spotfire and analyze it in the many ways you might want to. So when you've selected compounds in Spotfire, we have a display that tells how many different products you've made and how many different of each type of reactant was used to make those products. And if you're happy with your selections and with the amount of reactants that you're using, you're able to generate a fully combinatorial library at that point, where it takes all the reactants and uses them combinatorially. And this is what our production group, our library production group, will need. And so when this is ready, it's time to export the information back to KNIME. And once you do that, new nodes magically appear at the far right side of your KNIME workflow. And those nodes then contain the exported products, and you have a node for each of the reactants that had been selected. So the final steps in a library design then would be to do just a couple more things. One is to assign stereo category codes for each structure that's put in. We run a number of ChemAxon topology calculations and a few other magic things, and come back with a stereo code that'll be needed later on during the product registration. And once that's set, the library plan needs to be published so that something can be done with this, with your selected set of compounds. And the output of this is a zipped file that contains SDF files for each of the reactants and the products, along with the CSV file that indicates how many times each reactant was used in that library. And the value of this is that our parallel chemistry ELN can consume and parse that zipped file, and so this sort of completes the handoff from the design phase into the synthesis phase. So I finished going through the various features that we have here, and I hope that's given you a flavor of some of the things that we've done and ways that we've made things a little bit simpler for the users. The deployment was fairly straightforward. KNIME gets installed for everybody. We have a single DNS node update site that pushes out updates, and we trained users how to do that. In order to keep things consistent, we turned off the fairly frequent KNIME and Infocom updates, and so they don't get those updates automatically. But one nice thing is that we can take the very latest technology and run those on the nodes on the server. And so because that doesn't need to be in sync with the KNIME install that's on the user's desktop. Service updates, new models, new reactions, things like that don't require an update from the user's side. That's pretty much invisible. So they just open up the reaction browser, and there's 4 new reactions there, or there's new ROCS models or things like that. So In summary, all this work was done in about a 16-month period beginning in April of 2012, which was less than a year after the parallel synthesis group had been formed. We had a first release in June of 2012, and it was fairly readily adopted from the users. We got a lot of feedback from the users, and there was enough information or enough interest that people actually asked for additional KNIME training because they saw the value of doing other things. And we've had some phased releases since then, adding some new functionality, and at this point it's at a fairly stable state. So the only thing that's left is to acknowledge the many people who worked on this project. I'm not going to go through it all, but I will point out that we really had one person do nearly all of the node development, and that was Loki, who's here in the audience. He did all the nice icons that we had, and made a lot of very nice improvements for it. Okay, oh yeah, he's not here. He's not available, so yeah. And I'll thank you. Okay, and thank you for your time.
So a question. So you were mentioning ROCS, and I know I was looking at their implementation a few years ago, and you were mentioning the hours runs. They have now a CUDA GPU version that will take things in hours and do it in 6 seconds. I don't know if you've looked at it. I haven't yet myself.
Yeah.
Any negative?
Fast ROCS for this particular task. We're using it for other tasks.
Okay, so you like it.
Buy a GPU grid.
Okay, yeah, we're doing the same. Okay.
Any other questions for Brock?
Right.
So I got a question for you. In your workflow, you end up having reagents that get eliminated due to similarity. And medicinal chemists often think in orientation in terms of ortho-meta-para substitution. You may have then compounds or reagents that are very similar, but they are very different in terms of orientation that get eliminated in this process. So what are your thoughts in terms of overall approach of how you then go back and recover such compounds based on templates, based on substitution patterns, where linkers are associated, where linkers are located?
Well, that's a very interesting question. Really, the decisions about what to keep and what to exclude are driven by the chemists using these tools, and so so the workflow doesn't eliminate anything automatically. It's just, it provides information about what clusters things are in or calculated properties, and users make the decisions based on that.
Okay. Well, Chizzy, you definitely need a mic.
I'm just wondering if chemists are able to maintain or add new reactions, maintain reactions, or it's done for them?
We have found that it's that in most cases it's worthwhile doing a curated reaction, and so we do have a few chemists who have experimented with Reactor adding their own. Most of them aren't too comfortable doing that, and so over the course of 16 months they've only asked for 40 reactions, and so it's been fairly easy for me to put those in.
One more. Do you have a reactant database?
We do have a reactant database here. It is not attached to the system, although people can look up things from reagent numbers.
I'm sorry, I missed the difference between the bioreactor and the unireactor, and why you wanted to do it yourself and not use the Infocom node.
Well, the Infocom node does just the reaction, okay? And the other thing is that those have ordered input. So if the reaction is written with an amine first and an aldehyde second, then your amine has to go in at the top node, and the aldehyde has to go in at the bottom.
When you say that, yeah, your software detects
it sort of sniffs out, and we do actually use some of those nodes within the workflow that we have, but because we're executing the reactions on the server, we can add other nodes in that workflow. So it's not a one-to-one correspondence between an Infocom bioreactor and the DNS bioreactor nodes.
What is the difference between your reaction library and ChemAxon's reaction library?
Well, it's a lot smaller. There have been few libraries or a few reactions that we have taken straight from there. We do often start from the ones in there, but then we modify things to have a little bit more selectivity based on some literature examples. And we're not really trying to build chemical intelligence in there, so we're assuming that the users have picked their building blocks appropriately, so we don't have any of the exclusions in the reactions that you do. So nothing really gets excluded if it reasonably could react. Okay, but we do spend a lot of time working on selectivity, because they like to come up with things that are right on the edge with multiple groups that could react, and then you got to guess which one really does.
Okay, thank you.
Very good presentation. Thank you.